Sen U, or Hello Board of Down Syndrome Studies. My name is Joseph Dunne, and I am here today to release to you my theories about Down syndrome, with you in the hope you could offer me a grant to further develop my research and facilities. Okay, so let's get into it. Down syndrome is the most common chromosome disease in the world and is caused by having 47 chromosomes instead of 46, specifically a third copy of chromosome 21, hence why the disease is commonly referred to as trisomy 21. First described in 1866, the disease is very challenging on an individual and his family and occurs once in every 1,000 babies. The physical symptoms of Down syndrome vary between cases but are always quite significant and severe. Physical symptoms include a small chin, slanted eyes, poor muscle tone, large gap between big toes, and much more. As well as this, the average height of people with Down syndrome is significantly different to those without it. For males, the average height is 5'1", and for women 4'8". This then leads to other physical problems like obesity and instability in spinal joints. Down syndrome occurs on chromosome number 21, holding over 400 genes and 40 million base pairs. Unlike most chromosome diseases that are a result of mutation of the chromosome, Down syndrome occurs from there being a repeated chromosome in an individual's gene pool. The DNA changes that occur leading to Down syndrome is an error in cell division of chromosome 21. This is commonly referred to as non-disjunction. What this means is that during cell division, instead of one chromosome going into each new cell, both go into one, leaving the other daughter cell with no chromosomes. The reason for this error is still unknown, and a significant part of the funding I am granted would go into researching the environmental factors that influence this change. 90% of the non-disjunction occurs in the female egg, but I would investigate both genders to try to distinguish a pattern. Depending on how the DNA changes, different forms of Down syndrome become evident. The first and most common is complete trisomy 21. This is where all cells contain an extra chromosome 21. The next is mosaic trisomy 21, which is where not every cell has an extra chromosome 21. And then finally, there is translocation trisomy 21, which is where only part of an extra chromosome 21 is in the cells, not a full repeated chromosome. All types are affected by the different changes in the parent's DNA in the sex cells. In terms of being a dominant or recessive gene, Down syndrome falls into neither category, and is instead described as a chromosomic overdose, referencing the fact that it is not a trait as such, but a physical change in an individual's DNA. What this means is that the parents of an affected child's chromosomes reproduce ineffectively, leading to the child being affected. This is shown in my Punnett squares and pedigrees, so look close. Being in Mongolia, we currently do not have access to many screening facilities for Down syndrome, but for you Australians, diagnosing the disease has never been so easy and efficient. There are two main methods for diagnosing Down syndrome. Screening tests take place during the mother's first trimester and can see if a child is going to have Down syndrome. These tests are known for giving false results though. Diagnostics tests, on the other hand, such as chronic virus sampling or amniocentesis, are for mothers worried about their children having the disease, from screening results as a diagnostics tests are almost 100% accurate. The only negative though is they are very expensive. Currently, the treatment for most Down syndrome patients includes speech therapy, eye doctors, social workers, and physical therapists. All these practitioners are great, but the limitations include the fact they only assist carriers of the disease into fitting into the world. They do not cure the disease as such. With the grant, one of my many aims would be to establish a treatment method that could completely cure an individual of the disease. Biotechnology plays a massive role in treating and curing a disease, and that is evident in Down syndrome. Currently, the only biotechnology available is diagnostic testing to determine if a fetus has the disease. In the future, though, an aspiration of mine will be to create technology that can be inserted into a human fetus during early stages of reproduction that is able to eliminate all extra copies of chromosome 21. Funding for Down syndrome research is very important as the disease affects 6,000 people every year coming from all different racial groups. With funding, I would create a new biotechnology, discover why the manipulation of chromosome 21 occurs, and create a cheaper alternative to diagnostic screening so all women can afford to check their children for Down syndrome. Overall, I hope you deeply consider my presentation and take into consideration how much you would be helping the world by funding my research. Thank you.